Well, hello, everyone. My name is Robert Puckett. I'm an associate professor and extension entomologist in the Department of Entomology at Texas A&M University. And I'm actually delivering this presentation from the Rollins Urban and Structural Entomology Facility here on campus. And we do a lot of research on the management of very important urban pest insects. And of course, many of you know, if you're not new to this industry, that termites make up a giant proportion of the urban pest management industry in the United States, and in particular in the southern United States. So that's what we're going to dig into today. I'm going to address some novel understandings of products and application techniques, and some new information about termite biology in the United States. And I hope you'll be able to tease some information out of this presentation that you'll be able to use in your program, wherever you may be. So yeah, I'd like to take a minute and just thank Winfield for having me. I've given lots of presentations for them over the years and really enjoyed doing so. And we'll look forward to meeting up with you guys in person somewhere down the road. But for now, let's dig into termites. All right, so one of the first points I often like to make about termites is that the number of termite species around the globe is actually very small when we think of it in relation to the number of insects that have been described by science. So if we were in a live group together, I would ask for a show of hands, how many people know how many insect species there are? And there'd be 3 or 4 people that attempt to guess at it. And usually they would fall short by like an order of magnitude. There's well over 1 million insect species, individual species around the globe. But only about 3,000 of those are termites. And they're grouped into several different families, but there's only 4 of those that are important to those of us that engage in the termite management industry in the United States. So yeah, they're actually not a very speciose group when compared to the broader classification of insects. And in terms of their taxonomy and systematics, they're most closely related to cockroaches, believe it or not. In fact, they are now in the same order as cockroaches. They've been recently placed into that order. So you're going to have to relearn your cockroach and termite taxonomy. But even though there aren't that many of them around the globe in terms of numbers of species, they're very, very important, both ecologically and economically speaking. They're a very expensive insect group, and we're going to talk all through that. So there's some interesting things about termites that make them sort of difficult to deal with. And one of the first things that we have to sort of confront as we think about them as a group, these are small insects. They're soft-bodied, pale in coloration if you're looking at the workers or the soldiers. And that's really a very important point, is that they have soldiers and workers and alates or swarmers and king and queens. These are referred to as castes. So these are groups of organisms in the same family, all of which have different jobs. Each job is, of course, essential to the success of the colony. And so as we think about termites, we have to think about the different castes within the colony to determine whether or not there's one caste or another that should be exploited as we attempt to manage them. And again, this is a concept that we're going to talk all through later on in the presentation. So I mentioned termites are ecologically very important. And so you can basically find termites in all terrestrial systems around the globe, except in our extreme cold systems. So here's just some examples. This is a Reticulotermes termite, like many of you deal with in your business. These happen to be, we refer to them in my part of the world as agricultural termites. And these can be distinguished by, these are actually soldiers. These are nusudiform soldiers. And rather than having big, strong mandibles like this dampwood termite soldier, these guys have a structure on their sort of snout with which they can spray a noxious chemical in defense of the colony. And then, of course, if you've ever seen any videos talking about the magnetic termites, this is an example of their mounds, their colonies, and then, of course, this giant subterranean termite colony in Africa. So they occur everywhere, and they're very important to the systems that they occur in. And in terms of this sort of their physical description, these insects, of course, as you know, have chewing mouth parts. And they eat cellulose. And we're going to talk about this a bit, different points throughout the presentation, but that's all they're after as a food resource. And cellulose is a component of all plant cells. So this should give you a clue why they so preferentially feed on wood, because they're trying to extract the cellulose out of the wood. So they don't have a pupal stage. They molt through simple metamorphosis. In terms of wings, they've got 4 membranous wings that tend to be the same size, all 4 of the wings. And these occur in the primary reproductives, the alates, the swarmers that your customers call you about. And their antennae, so undoubtedly you've had a customer that's freaked out thinking that they were looking at an ant, or excuse me, a termite in their house, but in reality they were looking at an ant. Well, the antennae are one very, very important morphological character that separates ants from termites. Ants, of course, have elbowed antennae with an articulation point along it. Well, termites do not. Their antennae look kind of like a string of pearls, little beads. And they're broad-waisted too. So that's the other point to make in terms of distinguishing these from ants. They have a broad sort of lack of constriction between their thorax and abdomen, whereas, of course, you guys know, you think about an ant, we've got a big bulbous gaster or abdomen and then a tight constriction between the abdomen and the thorax. Okay, so we're all aware, termite control is a major focus of the pest control industry in most regions of the United States. I mean, that's why I've been asked to give a termite presentation for you guys. That's why you're seeking continuing education units related to termite work. But in nature, we think of termites as being beneficial insects. So the point is, you know, they're out there decomposing dead cellulose material, returning those nutrients to the soil. In a forest, right? Imagine a tree branch falls, hits the ground, or even a tree dies and hits the ground. You think about some of the trees in the western United States. These things are thousands of years old. I'm thinking about the giant redwoods. And those nutrients that are locked up in those thousands of years old trees are very important to the proper functioning of the ecosystem. So they have to return those as quickly as they can to the soil, or those nutrients have to be returned to the soil. And termites and other decomposing organisms are very, very important in that work. The problem is we can't train our termites to recognize the difference between a tree that falls in the forest and a 2x4 that's supporting a house, right, or some other wooden structure. So they don't know the difference. They're just feeding on wood as far as they're concerned. So as a result of that, they cause a lot of damage to man-made structures. And of course, that damage supports our industry, right? And as I mentioned, they're present throughout most of the United States. The other thing that makes them difficult as a population to manage is that they can live very long periods of time. So queen termites, subterranean termites, I mean, we've got, there's laboratories in the United States that have termite colonies that are, 12, 15 years old. So they're actually fairly long-lived as a colony. They've got an unlimited supply of food, right? So we provide them a lot of food in the structures that we build with our building materials, but in nature, there's, there's almost immeasurable amounts of cellulose that are available to these insects. And they live underground. So this is very important too. So they're protected from environmental extremes like heat, cold, flooding events, this sort of thing. And another challenge that termites present to us is that we have a variety of species in the United States and different types of termites. So if you think, we've got our subterranean termites, our drywood termites, dampwood termites, and many different species within each of those groups. So just like any other, and I know some of you guys are going to roll your eyes when I say this, but just like any other group of insects, maybe the most important ability for you guys to master is identification. And identification in termites can be really tricky. So I'm going to kind of walk you through some key diagnostic characters for identifying several species of termites as we work through this presentation. If you've not used identification keys before for identifying insects or plants, it's a good idea for you to get familiar with doing so. So there's lots of keys available for identifying different insect groups and in particular termites. So I'm going to give you my contact information at the end of this presentation. If you're ever interested in an identification key for termites, I can point you in the right direction. I can send you a PDF of a very good one. But if you haven't used keys before, they're kind of tricky. So basically it's a written description or a photographic or a photographic key. in which you take your specimen, you identify characters on your specimen that the key's asking you to find, and you compare the characters on your specimen to the key, to the clues that you're being given. And these usually occur in couplets. So two sets of characters are described for you, and you determine which one your specimen most closely matches. And so you get to the end of that couplet, and you're either presented a name or a number. If you get a number, well, you go to the next couplet. You go to that couplet number, and then you start to look at those characters that are being described. But if you get a name, you're done, right? You've reached the final step of the identification process. And then, of course, it's good to go get online and make sure things match up to pictures that you see online. And let me just walk you through a key here. This is a key to termites, of different families of termites in the United States. And so let me just kind of work through a couplet here, and I think you'll see what some of the challenges are when you're using identification keys. So couplet number 1 here, you can see is numbered, and we've got 2 sets of character descriptions. So anterior of each wing with 2 heavy veins. And so you look at your specimen, and turns out, I may have 3 heavy veins. So you keep reading, and it says, well, the fontanelle is usually present. Well, you look for the fontanelle on your specimen, you can't really see it well. So you just kind of keep moving, and you get to clypeus. Your clypeus is bilobed or divided by a line. And let's imagine you think yours is not divided. So what we're going to do is go and read the characters in the second part of this couplet. The anterior of each wing has 3 heavy veins, and actually yours does, your specimen does. And the fontanelle is absent, or the clypeus is simple, and yours matches those. So then you would go to couplet number 3, and we refer to this as a terminal couplet, meaning your specimen is going to match one of these groups of characters that are being described, and you're going to find your identification to family. And then the other keys that you'll find online in particular are picture keys. So, you just take your specimen and you kind of work your way down and you match it to this guy. And let's imagine your specimen doesn't look very close to this one. So you go to this side, and yeah, it turns out mine looks very similar to this picture. And then so you'll go to the next set of pictures and work your way through the key that way. But there's some big challenges here, right? So some of those may have occurred to you already. First of all, you have to know how to use the keys. Well, I've just described how you use the keys. That's not difficult. But you have to know the characters that are being evaluated. So if you were a live audience, we were there together, I would say, show of hands, who knows what a clypeus is? And most times, very few people do. And that's the problem. So when you first use a key, you spend more time like in a glossary of terms or getting online and looking up the names of the characters that you're supposed to look at and try to find on your specimen. It becomes very labor intensive. And then the other thing is the characters on your specimen may look very different than a picture or drawing. And the example I always use to describe this phenomenon is like if I hired you to build a photo key of termites, well, you would go collect species of termites, and I assume you would try to get the best photograph you possibly could, right? With the most perfect lighting, the specimens are arranged in the perfect way so that you can see all the characters. But the problem with that is that, the specimens that become available to you guys that have been sitting like on Mrs. Jones' windowsill for 3 months, and they're bleached out and only have 2 legs and the wings are broken off and they've been eaten partially by other insects. I mean, they never look like the specimens in the photographs is the point. So that can be really challenging. Yeah, and then of course the arrangement of the characters is very important too. So keys can be difficult. And the other part of that story is that, so this is a previous student in our lab, Dr. Danny McDonald. He's working with an ant key, but he's actually looking through a $12,000 microscope, right? So we use very good microscopes in our work, and they're very expensive. And so as a result, they're cost prohibitive for most companies to have them at their disposal. But that game is sort of changing. Obviously, you can use handheld, magnifying glasses and these field scopes and this sort of thing. They provide pretty good magnification. But if you need to see a really tiny character on a tiny insect, you need a microscope. And so this is, a bit of an advancement in the way we think about telling folks to get specimens to us or get photographs or to identify them themselves. So this happens to be a USB microscope. And if you've not seen these, there's lots of manufacturers that are coming onto the market. This one happens to be Celestron. I'm not selling microscopes for them, but this is the one that I have. I keep a couple of these around the lab. And you just connect them to a computer, either a PC or a laptop. And I think, you can appreciate the level of resolution we're getting there of old Abe's eye, right? So here's the other thing. I have a lot of PMPs from all over the United States that lean on me for identifications. And many times they do so from the field. So they'll take a photograph, with their cell phone and send it to me. Well, it turns out, like cell phone cameras are really good for taking photographs of like, large objects or a big group of people. But when you try to take a picture of a very tiny insect, that's where they fail. Unless you've got one of those macro lenses you can screw onto them. But the point is, cell phone cameras usually don't take very good photographs of small insects, and whoever you send them to is probably not going to be able to identify your insect from your cell phone photographs. I mean, every once in a while we can, but most times, we just need the specimen. But with this technology, these scopes can fit in your glove box. I mean, you can hook them to a laptop if you happen to carry one in your work, and you can get me a photograph, and maybe I have to ask you to turn it on a different angle to get a picture of a different character or something, but I can see them from these photographs. And so I've really been pounding my fist about these. Oh yeah, most important point of this, that's about $100 device. So they're very inexpensive compared to conventional microscopes. And you can get extraordinary resolution. So anyway, yeah, let's dig into the biology of subterranean termites. And so you probably sat through lots of these kinds of talks, maybe even today. We tend to arrange these in the order of, well, let's talk about the biology, let's talk about the identification, let's talk about management, right? And there's a reason why we do this as educators. I mean, I wish I could ask for a show of hands, but ordinarily I would just ask, well, how many of you guys are deer hunters? Down here where I am, deer hunting's big. I'm sure it is where most of you guys are. And you get lots of hands raised. And I'll say, well, how many of you guys consider yourselves to be an expert on white-tailed deer biology? And usually not a hand goes down, right? Because people have done their homework to understand the animal that they're hunting. Well, that's a perfect analogy for pest management, right? You really have to understand the biology of the organisms that you're trying to destroy to be able to do so, to be able to get chemical insecticide to the areas that we would predict they would be harboring in, to be able to predict their movement within a structure. It's very, very important to understand the biology of the insects that this industry seeks to control. I mean, it's fundamental. So anyway, let's talk about termite life cycles, right? And so obviously you're looking at this slide and you're thinking, well, what's the bed bug life cycle have to do with termite life cycles? Well, actually not a lot, but I'm going to illustrate a point here. The bed bug life cycle is very straightforward. They have no pupal stage. The nymphs hatch from an egg and they start through their developmental process, right? And they've got 5 larval instars that they're going to develop through and molt each time. They're going to take a blood meal at each life stage. And eventually, if all goes well for them, they're going to become an adult. And once an adult, male or female, they're going to look for a mate. They're going to mate. If it's a male, he's likely going to die soon. She's going to live on and lay eggs. If all that goes well, then they start the cycle all over again, right? That's a very straightforward life cycle. Bed bugs are a little bit difficult to manage too, but not because of their life cycle. Termites, on the other hand, have a very complicated life cycle. And this actually contributes to their our difficulty in managing them. And so we start here with the king and queen, right? So your customer has called you, or you happen to have witnessed a termite swarm event, right? So all of those individuals are seeking a mate. And if they find one, they'll pair off, and the female will lead the male around, and she will seek a place to start their colony. And actually, it's important to mention, so in most insect species, males are not very important after the mating event has happened. Usually they just kind of cruise off and die somewhere. But with termites, they're actually very, very important for the establishment of the colony, and then the maintenance of the brood and the maintenance of the queen over the course of her life and the life of the colony. So there actually is heavy paternal investment in termite colonies. This is really unique among insects. So anyway, they mate. She lays eggs, those hatch, and now we've got these larvae that begin to develop through. And if they become pre-soldiers, that's it. they're fated to become adult soldiers, males and females. And so, of course, soldiers are the defense force of the colony. They protect the colony from insects that might raid them. Some individuals become workers, and some of those workers just spend their whole life foraging for food and taking care of nestmates. Some of them, though, develop to the nymphal stage, and some of those nymphs become swarmers and swarm out sometime in the future and leave the colony to go start a new colony. But some of those workers can become, they develop through their nymphal stage into what we call neotenic reproductives. So second form reproductives, as you see here. And some workers just go straight to this reproductive form. And this is really interesting and complicated, and it's actually really important from a management standpoint, because these neotenic reproductives, those that became reproductives from the worker stage, the worker caste, they can lay eggs that become new termites, right? And that can be in the absence of a queen. Imagine a portion of the colony is cut off from the queen, or when the queen dies. There's individuals that continue to become reproductively capable. They can push out workers. And this happens when we go to the field and collect termite colonies. Oftentimes we don't get the queen. But after several weeks, we have individuals in the colony that are producing, that become reproductive. So it's very unique and sort of isolated to social insects in general. Okay, so I mentioned termite castes. These are the forms that can be present in a colony. We have the larvae, as you just saw, the workers, soldiers, nymphs, swarmers, and of course, king and queen. And the composition of these, the ratio of these is regulated by the queen and her pheromones. Okay, so swarming. So this is an event that your customers will not ignore. Now, they may ignore subterranean termite shelter tubes for years around their house, but when they have a swarm event inside, this does not get overlooked. And if they have the resources, financial resources to look into getting that solved by a professional, they're on the phone, right? Their hair is on fire because they've discovered a termite infestation in their home. And they have good reason to be, right? One thing I should mention, termite colonies don't push out reproductive swarmers until the colony is mature. And for most of our species of subterranean termites, that might be 5 years. So you've got a 5-year-old colony of termites feeding on a house. We're talking about a lot of worker termites that are removing wood as the clock ticks. So anyway, this swarming process is the process by which new termite colonies are started, and actually the mixing of genes throughout the population of termites in a given area. So these swarms are species specific to specific times of the year. So down here where we are, we've got lots of different species of termites. We get swarming. I mean, I get calls about swarming events happening in South Texas in January quite often. And we'll have swarming through mid to late October. And then we have several different species, as I mentioned. So the timing is important. Our retics, our native subterranean termites, are going first. And then our Formosan termites, the invasive species, move around Mother's Day. And then, of course, the dry woods are spread out across the year. Swarmers aren't strong flyers, right? Most of you know this. They're sort of at the mercy of wind patterns. And as a result of that, very few termites make it through the swarming event and successfully mate and start new colonies. Thank goodness. But enough of them do that it supports this massive industry in the United States. So what are they looking for after they swarm? Well, they're looking for nesting sites. And they have two basic needs. Now, there's exceptions to the rule, but their two basic needs are soil and moisture when we're talking about subterranean termites. They require a constant source of moisture. And this is why it's so important to communicate this to your customers. If you've got a subterranean termite colony that's foraging on a house, or in the case of Formosan termites, have moved into the structure, they've found moist wood. So whether there's been, an irrigation event that went sideways, a flooding event, a drippy faucet, a pipe, something is making the wood wet enough that those termites can forage on it and select it as a food resource. So you really need to communicate this to your customers, because you can kill them, but if they don't solve that problem, a few years down the line, if you use a conventional insecticide, a contact insecticide, that's going to break down. And if they haven't solved their moisture issue, they're going to be calling you back because a new colony will have found their house. Oh yeah, shelter tubes. So shelter tubes are designed, are used by termites to conserve their moisture balance as they move from the soil onto a structure. It also protects them from natural enemies and pathogens and this sort of thing. But as you know, termites move up and down the shelter tubes into a structure. And then with our native subterranean termites, they're returning to the colony with food resources that they've stolen from your customer's house. And they'll share that with their nest mates. With our Formosan termites, it's a little different story, and I'll get into that here in a minute. But as I mentioned before, moisture can be attained from lots of different things: leaky roofs, leaky pipes, shower stalls, condensation on pipes, humidity, you name it. Okay, so colony growth, colony development. So I mentioned before, a mature termite colony, well, it takes some time to reach maturity. And as I mentioned, yeah, it may take 5 years, depending on temperature and food resources and other resources in their environment. But it takes several years. And a mature colony, like I said before, is one that has all of the casts in it, at least for a portion of the year. Obviously, when the swarmers leave, there's no more swarmers in that colony, but they were there, right? So that is a mature colony. And depending on species, you might have upwards of 1 million termites in the case of our Formosan subterranean termites. Most of our native subterranean termites, you're talking about a couple 100,000 members. And I stressed this earlier: cellulose is the food source for all of our termites, right? And cellulose, again, is a component of all plant cells. So, with our subterranean termites, they're looking for wood in a structure, and typically they have to leave the soil to get to it if you don't have wood to ground contact. So in a slab foundation, they're building shelter tubes up the slab, finding a way in, either in a crack in the mortar between bricks or through the weep holes, or through cracks in the foundation. And this is much more difficult to resolve, but we'll talk about this in a bit. One interesting aspect of termite biology is that they don't have cellulase. They don't produce cellulase. And this is the enzyme that's required to break down cellulose as it's digested. So they have to employ the use of these gut microorganisms that live in their hindguts to help them break down cellulose, which is interesting. A lot of people have put a lot of research into determining whether or not we can kill subterranean termite colonies by basically treating them with an antibiotic, by killing their gut microorganisms. And the answer, the reality is that you can in the laboratory, but in the field, it's much more complex. And so I don't know, any of you enterprising folks that are research oriented, if you want to figure out that puzzle, you'll probably be able to sell that technology for lots and lots of money and retire. But anyway, the one thing to mention is they lose those microorganisms every time they molt. They have to get them back. And the way they get this back is kind of gross. Termite workers will regurgitate food for their nest mates. So that's one way they share their gut microorganisms. The other is by feeding on anal secretions from their nest mates. It's kind of nasty business, but they make it work. So, the workers in the colony, their job is to tend the other individuals in the colony, help them groom them, but then also to leave the colony and go forage for food resources. And they're not very selective in terms of the type of wood they'll eat. Pretty much any wood that's available to them is at peril for termite consumption. And that's even exacerbated to a bit when we talk about our Formosan subterranean termites. So this short section of this talk is for those of you that find yourself working in areas of the United States that are known to have Formosan subterranean termites. It's imperative that you can identify them from our native subterranean termites, because we're going to approach them quite differently in terms of managing them. Okay, so these are, they're not as widespread as our native subterranean termites, but we think of them as our most destructive termites in the United States. So they got here from their native system in Southeast Asia. They were recorded in Hawaii, in the Hawaiian archipelago, but this was back before Hawaii was part of the United States. And from there, they made the jump to the southern U.S., and it's pretty clear that they came along, they sort of hitched a ride in wooden articles that were returning from the Pacific theater of World War II, so post-World War II. And they made their way in and have been spreading since. They belong to the same family as our native subterranean termites, but they have quite a lot of different aspects in terms of their behavior and biology and management approaches. So one of the first things to mention about these guys is they don't need constant contact with the ground. Now, they will build shelter tubes and move from the soil into a structure and feed and return to their colony, but they don't have to. And I'll talk about that a little bit more here in a few slides. They have a giant reproductive potential, much greater than that of our native subterranean termites. They're known to attack a wider variety of materials, but just keep in mind, most of our building material is southern yellow pine. So, and both the natives and invasive species are happy to feed on that. Okay, so a big deviation in terms of their behavior and biology from our native subterranean termites is that Formosan termites will and do infest living trees. And this is a big problem that I'm going to talk about shortly. And they tend to have a greater tolerance to soil insecticides, our conventional non-repellent contact insecticides, as well as borates, as we're discovering. So in Texas, we've got them out in 32 counties, some of our most human-populated counties in the state. So think Houston, Dallas, Austin, San Antonio. Not yet out to El Paso, but some of our biggest cities have Formosan termites. Oh yeah, and so does the Bryan College Station area. In fact, I have a neighbor, one house back and 2 houses over, that have an ongoing infestation of Formosan subterranean termites. So believe me, I'm inspecting my house quarterly. Yeah, so how are they moving around? We've already mentioned that termites are pretty sorry flyers in terms of their nuptial flights or their swarming events. Well, we're moving them, we're doing them a service and moving their colonies around the United States. And one of the key culprits in this are our railroad timbers that get used. The used railroad timbers often get used in landscaping, so landscape timbers. They get reused, I should say. Another issue is mulch. So Formosan termites swarm at night, basically at dusk into the night, about an hour or two, and they're attracted to lights. So if you picture any of your big box stores that have a gardening area, where do they keep their mulch? Their mulch is usually stacked up on a pad out in the parking lot, kind of a roped off area underneath lights. And so we know that Formosan termites will swarm to lights, and we've got lots of situations like this. This is a bag of mulch, and I think you can pick out the shelter tube that's emerging from the mulch and moving under the baseboard, another portion of the garage in this case. So this is an instance where Formosan termites were brought into a house in a bag of mulch. So we caution folks around here to be thinking about this in the spring when these guys are swarming. Okay, so in terms of identification, distinguishing these from our native subterranean termites, it's very easy, especially if you can find soldiers. And actually, there's lots more soldiers in a Formosan termite colony than soldiers are in our than the number of soldiers in our native subterranean termites. And the big key diagnostic character is the shape of the head capsule of the soldiers. So you'll notice in the Formosan soldiers, they have kind of a teardrop shaped head, whereas all of our native subterranean termites have a head that's shaped like this, sort of rectangular. In other words, they've got long parallel sides on either side of the head. I think you can make that out. And as I mentioned, the Formosan soldiers are much more abundant. So with our native subterranean termites, sometimes if you break open a mud tube, you got to wait a while before soldiers show up, but not with Formosans. They actively recruit to the site of disturbance and try to defend the colony. And they're really, really aggressive. And they have this gland underneath their head capsule. If you disturb them enough, it's really interesting. You'll see this little glob of white material show up on their head. They're actually forcing that out of their head capsule. It breaks their head capsule, so they're doomed at that point. They're going to die in a while. And that material is really sticky. It's very tacky. It actually looks and feels very, very similar to like Elmer's glue. And if you touch it, they'll get stuck to your finger, which is really kind of comical. You got this soldier hanging from your finger, kind of kicking around. The point of that is that material is of no consequence to you and I. But now let's imagine you're an ant, and ants happen to be the major natural enemy of subterranean termites. So now you're an ant, and one of these soldiers has globbed you up with this big glob of adhesive, and now another one does it to another part of your body, and you're struggling to move around. Well, the next thing that's going to happen is that other soldiers are going to come up and start tearing you apart, legs and antennae and mandibles and everything, to shred you. So it's a very effective strategy for dealing with ants that raid their colony. So that's how you determine the difference between Formosans and our native subs in terms of soldiers. If you happen to have alates or swarmers, it's very easy to tell them apart too. So this is a Formosan subterranean termite wing, and all those little dots are meant to signify hairs. So our Formosan termites have hairs on their wings, tiny hairs, but you can see them with proper magnification, whereas our native subterranean termites are hairless on their wings. So that's the big separating point when you have swarmers. Okay, so the other thing Formosans do is they build a nest. So you're looking at what we refer to as carton nest. This was taken from a wall void. It's constructed of chewed wood, saliva, and feces from the colony. And those are tunnels that you can see in there. And they get really big, several cubic feet. And many times they're found above ground. They will build these in the soil, but from a structural standpoint, this is a dead giveaway that you've got Formosan termites. If you find these in a wall void, in an attic, in a basement area, you know that you're dealing with Formosans. Yeah, and another point to make is that they'll build these in the trunks of trees that they've hollowed out through their foraging. And that you can imagine this presents some big problems we're going to talk about next. But like I say, if you find carton nesting in a wall void, you are dealing with Formosan termites or a closely related invasive species that's now in the United States. I'll tell you about in a minute. So this is a tree that's infested with Formosan subterranean termites. This is a problem. You can see this colony has been working on this tree for a very long period of time. They've hollowed out the center of the tree and replaced that hollow void with carton nest. And that's going to continue to grow. Well, carton nesting is very brittle. It doesn't provide any structural support. So as that cavity grows, eventually, like the tree is still healthy physiologically speaking. In other words, nutrients are flowing in the vascular system of the tree. It flushes leaves every year. They fall off in the fall. But eventually, that cavity is going to be so great that the tree is going to lose its structural integrity and they fall. This usually happens in the spring in association with straight line winds and storm fronts. And you can get on the web and find tons of pictures of this. Trees that were infested with Formosan termites and have fallen on houses, on cars, on people in some cases. So it's a big liability. And a lot of municipalities choose to just lop those trees off. This was at a park in Baytown, Texas. I think they felled 27 trees that were infested with Formosan termites when we were down there. It was a big operation. These are two former members of our lab, James Austin and Chris Swain. I love this picture. This photograph reminds me of like deer hunters that got a big trophy buck, right? Makes me laugh every time I see it. But they were down there on the day those trees were knocked down because of Formosan termites. So you can see this is a big problem. Another story that you may be interested in hearing about, I hope you're interested anyway, is that we have another species of invasive termite in the same genus as Formosan subterranean termites. Coptotermes is the genus, and that is Coptotermes gestroi. This is the Asian subterranean termite. This is Formosanus. This is a Formosan subterranean termite. And if you look at the global distribution of these two species, there's only a couple places in the world that they share a common distribution. And one of those places is Hawaii. Hawaii, but also more importantly for the contiguous United States, Miami. And so there's a really well-known termite and ant researcher down in South Florida, Dr. Thomas Shuvank. He's at the University of Florida. He's stationed in Fort Lauderdale. And he was studying the swarm times of these two species. And what you're looking at is a phenology graph. So swarming activity on the dark gray bars here are the Asian subterranean termite. And the light gray bars are Formosan subterranean termites. And here are the dates that we're looking at, mid-February through mid-June. And what you can see is that there's a lot of overlap of these very closely related termite species. So Thomas wondered, well, I wonder if they can hybridize. And so he collected swarmers of both species and built crosses of these in a laboratory environment. And you are looking at the first photograph of a hybrid colony of Asian and Formosan subterranean termites, complete with eggs, larvae, workers, soldiers, mom and pop. Okay, so it's clear that they have the ability to hybridize. And what's most concerning about that is that if you, so what you're looking at here is a graph of the average number of colonies after per time, right, per unit of time. So this goes out to 200 days, and what you'll see is these are the crosses of parents of one species, right? So Formosan mom and pop, or Asian subterranean termite mom and dad, compared to the hybrid crosses. So what we're saying is the hybrid colonies grow faster. That's a problem. And the other problem is that they're more cold tolerant, meaning we expect them to be able to survive further into the interior United States than our Formosan subterranean termites or Asian subterranean termites. So keep your eyes on this. This is a developing story that we're all sort of tuning into and thinking about and trying to monitor, at least Thomas is in South Florida. So enough with termite biology. Let's move into thinking about termite control. And I the title of this talk is, News from the Underground, Novel Approaches to Solving Termite Issues. And this is a group of insects for which we don't have a tremendous amount of annual novelty, right? We have some big leaps in technology that occur many years apart. So I'm thinking now about, like, we went a long time in our industry's history of managing termites using repellent insecticides. And then, of course, the big innovation of non-repellent contact insecticides hit the research community first and then the marketplace for management of termites. So this was a big game changer in managing termites. And then a few years later, of course, Dr. Nanyao Su and Dow, he's at University of Florida, a termite researcher. They developed the first bait stations, the Centricon system, the first baits available on the market for termites. And then we've gone a long time since then to present. And there have been some significant advances in termiticide chemistry. Things like borates have come onto the market for pre-treatments and post-construction treatments. And then there's been some advancements in bait technologies, but these are sort of, we're expanding along the edges, the boundaries. We haven't made a big leap in technology in some time, but I am going to share some of the newer devices and pesticide chemistries that we're looking at in the research community, and some of which have already made it to the market. So before I do that, I want to introduce you to Dr. Phillip Schultz. He recently left our lab. He did his PhD with us, but he was also a full-time research assistant. And so Phillip was very, very instrumental for many years in executing a lot of our laboratory and field research. So we work as a team to develop plans, and then we work together to set up the projects. But Phillip was instrumental in following through and monitoring those projects through time. And he was very, very good at termite work. He worked as a technician for a pest management company down in Houston for many years before he came back to school for his graduate degree. He's now at the USDA, and of all things, he works on ceratopogonid midges. That was the focus of his PhD research. It's a midge that vectors an important disease of white-tailed deer in the United States, blue tongue, and EHD is the disease that they vector. Also a very important disease in production livestock. So we're happy Phillip has moved on to very important research in a whole different system. But anyway, before he left us, he carried out a lot of projects that I'm going to describe here for you. So one of the things that is really important for us to think about when we think about the application of conventional termiticides, contact insecticides, is how those move through a colony of termites. And so what you're looking at is a scanning electron micrograph of a soldier termite. And if you're unfamiliar with how this works, we sputter coat the insect or the object that we want to look at with the scanning electron microscope. We sputter coat them with gold, a thin layer of gold, because it's very reflective in terms of the electrons that we bombard the subject with. This is why electron microscopy is an expensive endeavor. And then we bombard them with electrons, those bounce back to a sensor, and it gives us an incredibly resolved image of the subject. And what you're looking at on the right side there are, this is conglomerate of microencapsulated termiticide. And here you can see that material affixed to the hairs on the antennae of this soldier. So we allowed these insects to forage through treated play sand in the laboratory, and then removed them, freeze killed them, and then took them to the electron microscope. And so this is what that material looks like when termites have tunneled through the soil that you've treated with a contact insecticide. So the next step is they return to their nest or to their colony, and their nest mates begin to groom them. And that's what you're seeing on this video. They're not eating this termite worker, sort of the blue one here, that's been fed a diet that's been dyed with a blue dye, so we can pick out the donors in this case. So as they groom this insect, they're taking in molecules of insecticide. And then, remember earlier I mentioned they'll regurgitate their food resources for nest mates. That's what you're looking at here. They're feeding one another and passing along the active ingredient to their nest mates in that way. And eventually that material moves through the colony, kills enough workers that they can't support the queen, or potentially kills the queen herself, and the colony winks out. So let me show you some results. I want to make the point. I want to be very clear. Like I've never I've never earned money solving pest problems for customers, right? So I do that on the research side. So I've not assumed the risk of taking on customers like you guys do, but I do assume a pretty risky endeavor in that we trial insecticides and other products that we can then recommend to you guys. So we take that very seriously, because I know if I tell you something, you make a product decision based on something you heard me say in a talk and doesn't work, well, I've just cost you potentially your business's reputation. So I just want to make clear you guys understand how seriously we take this. And what I'm showing you are results from our field trials over many years, working with different insecticides, different termiticides around structures. And we are very, very precise in terms of how we apply these, how we assess the amount of material that we're going to put into the soil, and how careful we are when we mix termiticides in our tanks. And then we do the work just like you, right? We're trenching, we're rotting, we're drilling. And these are some of the products that you'll recognize: Ultraset, Fuse, HP, the term BASF product, HP2, Premise, Taurus, SC, Termidor, HE, etc. This is the number of houses. I think there's 61 homes that were treated here, but this is the most important column. In your world, you would call these callbacks, but these are retreats that we had to do over the course of these studies. 2 over the course of all those studies. The point I'm making is that we take the calculation of the amount of materials that we're putting onto a structure very, very seriously. And you should too. Because if you do it right, you can be very, very effective and very successful, drive your company's reputation through the roof, and help you compete with other folks who are doing termite work in your area. And, these are some photographs from some jobs related to research trials, field trials that we've done. This is just to illustrate that if you're thinking about getting into termite work and you think this is going to be a walk in the park, you are sorely mistaken. And the guys in the room, your colleagues that do termite work, can back me up on this. It is a labor-intensive endeavor. So this house has been trenched. This is concrete, of course, and we've had to drill through that every foot. That's a labor-intensive and time-intensive endeavor that carries with it its own liability, right? You crack somebody's concrete, you break a tile, what have you. You got an irritated customer. You got to learn how to do this work. And your customers have to understand that this is not like, we're not baiting, fire ants in a yard in the South, and putting out a broadcast application of granular baits. This is serious work. And as a result of that, it's going to have a serious price tag. This is why termite work is very expensive. And so some of the big product innovations in the marketplace now that you're starting to see show up have to do with eliminating man hours on the job site. And so if you're familiar with this product, the Termidor HP2 high precision injection system, all this device is designed to do is to eliminate man hours on the job for your company, and to be able to provide you a little bit better competitive bid for your customers. So the way this device works, it mixes your chemical, it applies the proper aliquot of chemical to the soil, and it forces it deep into the soil with pressurization. And I'll show you a video of this working. This is Dr. Bob Davis from BASF. He's using this device. It's turned on its side, so it's shooting horizontally. And I think you can see that liquid that it's forcing out. In this case, it's just water, but it's forcing it out under a significant amount of pressure. And had that been the soil, an appropriate soil type to receive it, it's going to force that termiticide deep within the soil. Now, this doesn't eliminate drilling. So if you've got concrete abutting a structure, you're still going to have to drill. But the days of trenching are over if you choose to use this product. I'm not sure what the price tag is now, but I do know they're very pricey. So you have to determine if you do enough termite work in your business to support that. But anyway, this is an example of pretty significant application device innovation in our industry. Sort of a new use for an old product is the use of borates for pre-treatments for wood-destroying insects in homes. So borates have been around and used as an insecticide for decades, if not longer. Since the 40s that we know of in this country, longer in other countries. We know that if you provide insects a source of boron salts that they'll take as a food resource, they'll die, right? It will kill insects. We've killed almost any insect that we feed borates to in the laboratory. So borates are used in the wood-destroying insect world, the structural insect world, as a preventative. And in some cases as a remedial response to infestations of termites. And so we've worked with borates too in our work. So Texas is a state that is allowing borate pre-treatments of homes. And if you're not familiar with that, it looks just like what you imagine when the home is framed up. A technician comes in and treats the lumber, the framing timber in the house with borates. The borates are absorbed by the wood. They penetrate the wood to significant depth. But the good news is they're locked there maybe forever, maybe longer than the life of the home. So that if termites discover that wood at some point in the future, that insecticide in this case is there locked in the wood. If they feed on it, they're going to die, which is, very comforting for homeowners. I mean, I might still think about using applying a horizontal barrier of a contact insecticide, because we know that's going to buy us at least half a decade post-construction in terms of protection from the house if the complete pre-construction treatment is done. But anyway, let me tell you about a borate treatment trial that we did in the laboratory. Phillip treated these boards with borates. I can't mention the manufacturer yet because this product hasn't hit the market. But then he stored those blocks of wood in a cabinet in the laboratory for 3 years, pulled them out, attached these very sophisticated research devices. This is an inverted plastic petri dish held in place by a rubber band onto that wood, and allowed termites to feed. So what you're looking at is the mortality response in these termites. This line is our untreated controls. Not unexpectedly, after 14 days, we had very little mortality among the controls. This is Boracare, and we did get significant mortality of the termites that were exposed to the wood. This is number of termites. There were 40 in each of those dishes. And so you can see we got pretty good control in terms of percent mortality of the termites. This mortality response relates to the low, medium, and high rate of this borate that was being trialed. And you can see we got very, very good mortality of the termites that were exposed to that wood. So stay tuned. There'll be more to say about that product in the future, but that's as far as I can go now. But I do want to talk about baits and how effective baits can be for baiting for subterranean termites. So all of our baits use an IGR, an insect growth regulator, as the active ingredient. And all these products do, these active ingredients, is prevents these insects from molting through to the next developmental stage of their biology. So if it's an adult that feeds on an IGR, it's an adult worker, it's going to die soon anyway, so forget about those guys. But the insects that are at this life stage just before adulthood, they're not going to become adults. Any of our members that are going to become swarmers that aren't quite there yet, they're not going to become swarmers. And as a result of that death, the worker load is going to be reduced in the colony, the soldier load and the swarmer load. And eventually, there's not going to be enough termites to support the life of the colony, and they're going to die. So the pros of using insecticidal baits for termites is that, it can reduce labor. Treatments can be faster than conventional insecticides. They can certainly reduce the chemicals in the environment in terms of termiticides. They can contribute to repeat business because you got to come and service those annually or more frequently. The cons, of course, is that foragers have to find the bait first for that bait to start killing them. And actually, they're spaced pretty far apart around homes, typically 10 feet. 10 feet is a big area for foraging subterranean termites to slip through your bait stations, but eventually a colony is likely to find them. And they're slow acting. So this is one of those areas where you really have to educate your customers. It's going to take a while for these to work, several months in many cases, sometimes much longer, but they will work. And I've got some data to back that up. I'll show you a field study that we just recently ran. A new product innovation in the baiting arena is the above ground bait stations that some of you are familiar with. Basically, the technology is the same. We've got a station that houses the baits, in this case, a dry pelletized bait that you add water to, and you knead it like a bread dough till it's soft. You put it back in the container, and we're going to apply those directly to the foraging tubes and areas of activity of termites on and in the structure. Basically, you're taking the bait, in this case, directly to the termites. These are some potential application areas for above ground bait stations. So anyway, these are coming onto the market, and they can be wildly effective. I mean, we've treated many buildings on our campus using above ground bait stations in product trials. And, in most cases, they're almost 100% in terms of death of the colony. So here's some data related to a couple of those. So we got two colonies of Formosan subterranean termites in different structures. They consumed, in one case, a lot of bait, 620 plus grams. In another case, only about half of that. Both colonies winked out. One took, between 60 and 90 days to do so, and the other within 30 to 60 days. And then our native subterranean termites, 2 colonies of those. And the consumption was comparable to the Formosans, and actually the time to kill of those colonies was very comparable as well. But the point is, they died. So these are effective. These weren't laboratory trials. These were actual field trials on living colonies. And this is a much bigger field trial that I want to describe to you using genetic tools to understand what goes on when we use bait stations in the field. It's really cool. So in this case, we're working with a BASF Traylona system. And this is a true field study in that these were not termite colonies associated with structures. These are termite colonies out in a wooded area and a pasture area, and combined, actually. So let me show you what that looks like. These are our 2 field sites. One was actually baited, and one was baited with a placebo. This was our control site. And you can see it's a mixed habitat, woodland versus open. This is an undeveloped site within about 400 yards of our laboratory that we gained access to. And this is a grid. Each one of those dots that you see at each of these sites is a site where we placed a bait station. And then we used microsatellite markers, basically a molecular technique for fingerprinting those colonies. It's essentially the same technology that's used in like forensics and paternity testing. And so what we can do is we can go into the field, put bait stations, none of which had active ingredients. We can service those monthly until they're hit. Once they are hit, we take some of those individuals out. We fingerprint their colony. We give them a name and number. Put the bait station, the placebo in the controls or in the treated site, actual IGRs, and then watch what happens to that colony through time. So what you're looking at here are both of those 2 field sites through 4 years, so 16, 17, 18, and 19. This was our control site, and you can see at the start of the trial, we had a number of numbered and named colonies. The number of colonies grew through time. And you can see many of these occur year after year. Now, they may be in a slightly different location, like this colony number 3 had moved over a couple of feet to another bait station from year from 2018 to 2019. But the point is they stayed and we could find them. This is our treatment site. And you can your eye can probably tell you as we move from 16 out to 2018, then to 2019, the number of colonies is diminishing. And the reason for that is because we started baiting them with actual hot Traylona stations. So if you look at this graph through time, you can see the number of colonies at our baited site versus control site were very similar. So the dark bars are the baited site, the light bars are the control site. And then just here in 2018, we put active ingredients into the colonies in the baited site. And you can see they start to diminish. And you can kind of see this same phenomenon on this graph. It's just a slightly different graph telling the same story. This is the control site. The number of active colonies kind of went up pre and post treatment, but that wasn't the case in our baited sites. They went down precipitously. And then this is just another way to look at this. This is the number of colonies in our treated site from 2018 to 2019. This is the duration of colonies at the control site. And you can see they spanned the baiting time and remain persistent throughout the time of the life of the study. Whereas in the sites that were baited, you can see there's the baiting event occurred, and then we start to lose those colonies. Now, some new ones showed up as a result of nuptial flights that landed in that treated site, but actually they winked out pretty quickly too. So the point of all this is that baits work. We know they work. The termites have to find them first, and that can take some time. And then the baits have to work on them in a different way, sort of than contact insecticides work on our subterranean termites. But they will work. It's just going to take a while. And that's up to you to determine based on the temperature of your customer, right? If some customers just don't want contact insecticides around their house. Awesome. Baits are your answer. And in some cases, they do a cost-benefit analysis, and you might still have to convince them, well, even though this might be a little less expensive through time, your bait stations are going to take a while, so they're going to have to be comfortable with the idea that you haven't fouled up. It's just that the whole strategy is different when we're using baits. So anyway, I'd just close by saying this. You got to be able to identify your termites. If you can't, you're lost. And there exists a pretty wide variety of approaches and products that can help you successfully manage your termite customers' problems. But you really got to educate your customers about how each of those products work, the perils and hazards and pros of using either one. Get them comfortable with what you think you want to do. Once they're comfortable and they understand the procedures, it's going to be a lot easier explaining them on the back end when you have a little bit of sort of persistent termite activity that you and I know are going to cease to be active at some point in the future. So, yeah, just mention if you ever need help identifying specimens, you can't get a photograph to me, you can very easily send me specimens. Just drop them in alcohol and something that won't leak. Contact lens cases work perfectly for this. Go to our website. We keep a form on the website that provides information for how to get those specimens to us. Please don't stick them on tape. I still harp on this because I still get them. Just like your customers find insects in their windowsill and put them in Scotch tape for whatever reason and hand them to you to ask you what they are. PMPs do that to us, and it's just impossible. You got to open the tape, and heads end up on the wrong side of the tape. If they're soft-bodied insects, they get smashed in the mail like these termites. These happen to be Formosan termites. Just don't do that. Drop them in alcohol. Send them to me. So yeah, here's all my contact information. If you have questions, I'll be here to answer them for the next few minutes. If not, yeah, just reach out to me through one means or another, and I'll be happy to work with you. Take care.